Hi, all. Dr. Clark here, and today I'm going to talk about the formal lab report. And normally I do this in a live session when you come to campus to do some labs, but under the current circumstances, we're not able to do that, so I'm bringing it to you in video format. The formal lab report is a large portion of your grade. You'll see by examining the rubric that's put forth. And it's a very important piece of your grade as part of the skills that you should gain from doing a biology course or a biology lab course. One of the main skills is you need to be able to write scientifically. You need to know what it means to do scientific research and how to present that research in written form. That's what the formal lab report's about. Okay? So the first part of the formal lab report is really on you. What you need to do is you need to select one of the labs that you have done or you will have done throughout the course, one of the labs, and you need to write a formal lab report on it. I suggest selecting one that has a fair amount of data that makes it easier to run statistics and to make graphs or tables. Okay? And really it allows for you to get at more data, more information, and makes it easier to write a lab report. So regardless of what lab you choose, that's purely up to you. The design of the lab report is not up to you. It's based on a rubric, and it's based on the normal format of a scientific lab report, a formal scientific lab report. Okay? So with that being said, we're going to jump into the pieces of a formal lab report, and I'm going to show you and share some information with you, some hints and some key components to each one of the pieces of that lab report. Okay, so first off, here's the rubric. I shared this with you. Okay, you know how many points each section's worth. Okay, so the title page is worth 2 points, abstract 7 points, etc. Introduction's worth 20 points, and this is how it's broken up: 10, 5, 5. Okay, so if you have a question like, wow, I didn't do a title page, how many points am I going to miss? Okay, or I didn't do an abstract, how many points am I going to miss? Okay, if I didn't do statistical analysis, how many points am I going to miss? Okay, so all that information is there. You know how many points each section is worth. So I suggest, referring to this when you're in question of, well, what do I need in that section? And also how many points is that section worth? Okay, so let's look at each one of these sections, these main sections, and I'll talk about the components underneath them. Okay, so the first one is the title page. So the title page is not worth very many points, but it's fairly easy and important to do. First of all, you need a title that describes whatever the study is that you're going to be presenting. Okay. You don't need to write chapter. This happens to be a publication that I wrote for a book. And so this was chapter 1 labeled there. But regardless of that, it tells you the title. Okay? It tells you what you should expect to find in that lab report or in that section of material. Okay, and then you guys can write this if you would like. This is to be submitted to, Western Wyoming Community College Biological Lab Sciences, Dr. Will Clark, or whatever you want to write, that you're submitting it to me. Okay, in this case, I needed to write who I was submitting it to. Okay, Environmental Biology of Fishes. Okay, it's myself and a co-author. But this is an equally important piece. As you can see, there's a superscript here by my name, okay, and it corresponds to a address or a way at which you can get a hold of me. Okay, you guys don't have to give me your physical address. That's completely fine. You can just give me your email or some way or form to contact you. Okay, there's only going to be one author on your paper. So that's just going to be yourself. And then your contact information, basically your email. If you want to add your phone number or your physical address, that's completely fine. But that's important to be on that cover or title page. So when I look at it, I know exactly who it is, because maybe this entire time you have not been using your formal name, like myself. And now you use your formal name, and I'm like, well, I don't really know who that is. I know that individual by a different name. Okay. That way I have an email contact for you if there's questions or whatnot. Okay. All right. Title page. Pretty simple. Not that many points. Simple to do. The abstract. Okay. So the abstract should not be longer than a single page. So here you can see, again, this is part of a book. So starting on 114 was this abstract. Okay, it is 1 page long. The key components to a good abstract is that you need to start off very broad. So in this case, it says parasites can negatively affect individuals' physiology, resulting in energy trade-off between the immune system and other life history components. That's really broad. It's not talking about what type of parasites. It's not talking about what type of physiology. It's not talking about what type of life history it can affect. Okay. Then you start to get more specific. Okay. So now we're talking about what's happening in this study, what happens in your research. So in this case, it then says, in this study, we examined relationship among parasite density, fish condition, fish immune function. Okay, so now you're telling them what you did. Okay, and then you're telling them how you did it. So kind of methods of how you've done it. Okay, once you get down in there, after you've done the methods, then you start getting into your results. So your abstract is basically an introduction, a little bit of introduction, maybe 2 sentences of introduction, methods, 2 sentences of methods, results, 2 sentences of results, okay? Discussion, 2 sentences of discussion, okay? That's it, okay? It's concise. It's basically an overview of all the high points of your entire paper. So when I get down in here to the methods, okay, I talk about statistical methods here. I say model selection based on information criteria rank. Okay, models included terms of at least one parasite taxa higher than models without terms of parasite taxa for most measures of condition or immune system. You don't need to know what I'm actually talking about, because for most of you this is beyond the scope of what you've learned in biology, but I'm talking about a statistical test that I did, okay, or a way to statistically analyze condition factors and immune systems of fish. Okay. Then later on, I tell you, okay, the next sentence, I tell you what the results were. Okay. So in this case, Necessica, that's a type of a species of parasite, had the highest infection rate, 25%, and occurred in the majority of models, explaining variation in the white sucker's condition and immune function. Okay, that's a highlight. That's basically saying, hey, look, if you have this one parasite and it's in 25% of all the fish collected, and it's explaining the variation that we see in the condition of the fish and the immune function of the fish. Okay? So that's important pieces. Now, you'll see that there's a lot more data and a lot more analysis as we progress through the study, okay? But I'm pulling out high points. Okay? And then at the end, you want to kind of tie it back to the big picture. In this case, I say, this study emphasizes how variation in life history traits across species, geographic range may be related to physiological factors that are related to parasites in fish. So again, I started off with, how do parasites negatively affect an individual's physiology? And then at the end, I say, okay, due to the results of this study, it seems like, indeed, that across all geographical range of this species of fish, parasites can have a negative effect on the physiological factors of this given fish. Okay? So again, abstract. You want to have intro, methods, results, and discussion. Okay? All right, so let's jump to the next page, and we're going to look at introduction. Introduction, this is what I would consider some of the meat of your lab report. This is where you're going to get a lot of resources, a lot of background information, and you're going to get it in here so people understand why you would have done this study. Okay? What's the importance of the study? What have other people found regarding this study? And then what are your objectives or your hypotheses of this study? So, introduction, you start off very broad. Okay? Extensive landscape alterations can disrupt the function of many biological processes, including individual physiology. Okay? Huge. That's really broad. Okay? Then you start getting into what does it mean as you get more specific. Okay? Changes to aquatic environments. So now I'm talking about aquatics, not extensive landscape. Okay? I'm talking about aquatic environments. Can have direct effects of growth, reproduction, survival in individual fish. Now I'm telling you, hey, I'm looking at fish. Okay, up here I'm just telling you that landscapes can affect organisms' physiology. Okay, now I'm telling you that it can affect growth, reproduction, and survival of fish, as well as it indirectly affects the predators, the parasites, the competitors, and food resources. Now, you can see here that I have a bunch of names and dates after this material. Okay? It's not quoted because, again, in biology, we don't quote anything. Okay? We paraphrase, and we take information from this guy, and we take information from this person, and we combine it all to make a beautiful sentence that basically states that changes in the aquatic environment can affect the fish's growth. Okay, growth came from McCrunchick and Dixon. Okay, their report was about growth. Okay, Lafferty and Curris, their report was about parasites. Boltel and Magnan, their report was about survival and reproduction. So what I did is I stole pieces, borrowed pieces of all these other research, okay, to basically state, hey, look, other people have found that if you change an aquatic environment, it can affect the predators, the parasites, the competitors, the food resources, which ultimately can affect the growth, the reproduction, and survival of fish. Okay? So start off very broad and then start becoming more specific. You're pulling from papers and information from other individuals to provide background evidence for why you might be interested in studying the material that you're studying. Okay? And so you can build on that. And then once you get super specific, basically into what you're going to look at in the given study, that's when you can start talking about hypotheses. Or in my case, I prefer objectives versus hypotheses because I don't use statistical tests to test hypotheses. Instead, what I do is I build models to explain big picture ideas, how parasites and predation and competition and food resources can affect reproduction. So it's multiple faceted, multiple pieces. And you don't have to worry about that. You can just stick hypotheses in because you're going to be examining a much smaller, more concise lab project, okay? Not a big picture idea. Okay, but at any rate, it's the same deal. You're going to provide background using resources, using scientific papers, scientific evidence, peer-reviewed journals. You're going to provide background for why it's important, what other people have found for that information, given other people's research, okay, etc. And then what you did for your research, okay? And you're going to have your hypotheses here. So in this case, I have objectives, 2 objectives: evaluate the relationship between parasite infection and lipid storage. Evaluate the relationship between parasite infection and immune function quantity using hematocrit counts, leukocrit counts, and neutrophil lymphocyte ratios. Excuse me. So here you could just have your hypothesis. Okay. Researchers believe that there is a negative relationship between parasite infection and lipid storage. Okay, there's your hypothesis. So you don't have to have an objective, you can just use hypotheses. Okay, that's going to come, your hypotheses or your objectives are going to come at the very end of your introduction. So here you can see my introduction is 4 pages long. Yours doesn't have to be 4 pages long. Just have enough information that you provide adequate background for the lab that you're doing, the lab report, the information, whether this be on photosynthesis, this be on cell cycle, this be on cellular respiration, this be on biomolecules, whatever it might be, whatever lab you choose, okay, you're going to provide adequate research, adequate background information on the processes by which that lab works. And then you're going to provide hypotheses for the results that you should obtain. Okay. All right. Moving on. Methods. Okay. This is where a lot of students get confused because I don't know if in high school or middle school or something, you have instructors that teach you to list your methods, like method number 1, 2, etc. But that's not how you write a method section. A method section should be written in paragraph form with, if there's steps, with the steps listed out, okay, in chronological order, but not listed as step 1, blah, blah, blah. You're just writing them out as first we did this, second we did that, okay, etc., etc., etc. But your method should always start with the study area. Where did it take place? Where did the research take place? Okay, where and when? So if you have exact locations of where it took place, that's perfect. You have exact dates and times of when the project was done, that's perfect. Okay, that comes first. Okay, so again, you're starting very broad. Date, time, location. Okay, then you're becoming more specific. Okay, so maybe you have multiple methods, multiple things that you're looking at. In this case, I was looking at collecting white suckers. Okay, that's a type of fish. Okay, in the field. So there's my field collection methods. Okay. Then after I collected them, then I need to get the condition of them. So then I took condition factors of those. Okay, so maybe you're looking at a lab report that has 4 main steps or 4 main things. So you're going to have methods for the first step. Okay, maybe you're looking at chromatography. Okay, then you're going to have methods for the second step. Maybe you're looking at photosynthesis in the dark. Okay, or cellular respiration. Okay, whatever lab you're choosing. Okay, maybe you're choosing biomolecules, and your first step is you're looking at proteins. So you're going to have methods for examining proteins. And the second one, you're going to be looking at lipids, and you're going to have methods for examining lipids. And then the third one, you're going to look at carbohydrates. Okay, you're going to have methods for carbohydrates. Okay, so you're going to break them out so people know, hey, look, I'm interested in proteins. These are the methods at which you have done to get at the proteins. Okay, these are the methods you've done to get at the lipids, whatever it might be. Okay, and then a really key component that a lot of people forget is after you've done all of those methods, you have data. What do you do with the data? You do statistical analysis on the data. So you have to have some statistical methods. So here you can see that I have spelled out exact statistical methods, model building, etc. that I've done with the data. Okay? And this, my method section is like 15 pages long. Yours, just as long as you need to get the methods across. So someone can pick up your methods and regurgitate your study. That being said, if you're using the methods I put forth in the lab manual and not changing them, remember, that's plagiarism. Okay? So you need to rephrase them, rewrite them, okay, and excuse me, and cite where you got that from. So, for example, you can see that I have citations in this paper, like this Fulton condition factor. Okay, this equation came from Ricker, 1975. He's the individual that did this. Okay, this statistical method came from our development core team in 2006. Okay, so not only are some of the methods not my own methods, okay, I use methods that are published. Okay? You're going to do the same thing. Okay? You're going to use methods that are published from the lab manual. So you need to cite the lab manual, okay, for those methods that are not common knowledge. Okay, common knowledge methods, simple things like grabbing test tubes and pouring material in test tubes. Those are all common knowledge. But what's being added to the test tubes, what the specifics are, okay, that's not common knowledge. Okay? You wouldn't have known how to do that prior to reading the lab manual. So really simple steps, okay, like putting on, gloves, or, steps that you might have in the lab. Now remember, these are all virtual. Okay, so your lab shouldn't say, I turned my computer on, went onto YouTube, and clicked play the video. Okay, you need to be actively imagining yourself doing this lab, okay, by using my video as your hands. Okay, so you're following the steps. Okay, and if you see me do something in the video that's not in the steps, okay, you need to write down that method that was done that's not in the steps in the lab. Okay, and I know it can be difficult because it's all virtual, but again, this is the day and age that we're living in and dealing with. So this is what you have to do. All right, next section, results section. Okay, some key components with the results section. You always start the results section with written information. Okay, this is normally just general information. I even wrote general parasites. So this is general information about parasites. Okay, and talks about, the number of parasites found in each one of these rivers, the statistical analysis, comparing the rivers, et cetera, is really basic general information. Okay, that's what you want to start with. Then you can get into more specific information, and you get into where you're going to put tables and figures. So if you can see here, after this little brief thing, this relation is most likely a reflection of the size differences between, individuals, infection rate being significant. Okay, and then I say C, or I have figure 15 here. Okay, that's telling the individual that I'm citing, hey, look, you should check out figure 15. This is referencing your figures. You need to do that. That's part of a lab report, is you don't just make graphs and tables and then not write about them or not reference that information. Okay, so you want to start off general and then become more specific based on what were you examining. So maybe the general would be something very simple, like, maybe you're examining photosynthesis and you're looking at really simple stuff like, we examined chloroplasts under the microscope. Okay, general information. Well, maybe we counted stomata. Okay, really general information. Then you get into more specific stuff, more complex methods. Okay, so maybe chromatography or something like that. Okay, so you need written results, and then you need figures. Okay, and tables or tables. Okay, so you need greater than 2, so at least 3. This is a table. You can see tables are, a list of information. Okay, in a format like this. They have rows and they have columns. Okay, this is how you need to write a caption to your table. Caption for tables go above the table, and you should number your table based on the order at which they show up in your lab report. So this happens to be table 21, happens to be the 21st table that was in this chapter. Okay, and then it tells you how to read the report. Okay, it's telling you that your columns, there are female, male, juvenile columns. It's telling you that there are independent variables. It's telling you if there's statistics, it's telling you about the statistical information. Okay, if you use any abbreviations, okay, they should be listed. So here you can see FAL is fall, and I list it, so I abbreviated it, fall. SPR is spring, I abbreviated spring. Okay, so if you're going to use abbreviations, they need to be present in your description or your caption of your table. Okay, these are figures, okay, sometimes called graphs. Now, graphs, there are lots of different types of graphs. You can do pie charts or pie graphs, bar graphs, which are actually called column graphs. Okay, you can do scatter plot graphs, you can do line graphs. There are lots of different types of graphs, but they're considered figures. And so when you label them, it should say figure and then whatever the figure number is. Okay, and then the description of the figure. Again, if you're using any abbreviations, etc., you need to include those. The figure caption goes below the figure. Okay, so the table caption goes above the table. The figure caption goes below the figure. Okay, make sure that you've labeled the X and Y axis. Okay, and then if you need to explain how to read the figure, you need to explain how to read the figure. In this case, you can see that I have doubled figured this figure because I'm limited on how many figures I can have in a given chapter, so I combine these. So this one is percent lipid in liver versus the density of Necessica. This is HSI or hyposomatic index, okay, or hepatosomatic index, and density of Necessica. So it's the same x-axis, just switching the y-axis, and you can see the results. Okay, I'm not expecting you guys to get all crazy, but I'm trying to explain these figures to you. If you read here, it will tell you, okay, that is what's going on within this figure. Okay? Here is a normal column graph. Again, it's a lot more complex than I'm expecting from you guys. Okay, this is showing parasite percentages of all the different parasites, okay, in one figure. So you can see from these are different fish sizes, okay, and the parasites that are present. Okay, again, caption underneath indicating that. Okay, so if the figure is confusing and you have no caption, then no one can read it, no one can understand it. Okay, so that's what the caption's for. It's to clarify information about the figure that might be in question, like abbreviations or that you have two figures or that each one of the bars means something different. Okay, that kind of information needs to be present. Okay, so that's the results section. Again, you want to start off very general, become more specific, make sure that you're citing or drawing reference to your tables and your figures within the written text. Okay. All right, discussion. Equally as important as all the rest, and often people, by the time they get to the discussion, this is where they're like, I'm done. This is a lot of work. This is not fun anymore. Okay. The discussion is where you're going to discuss the results that you figured out. Okay. So this is where you get to add your opinion based on the data that's collected. That's one way. So remember, I discussed this before. There are two ways to use sources in biology. One is to base your opinion on data that you have collected. Two is to base your opinion on data that others have collected. You're going to use both in your discussion. So you're going to discuss your hypotheses, specific hypotheses. Are your hypotheses or your objectives, did you support your hypothesis? Does the data support your hypothesis, or does it refute your hypothesis? Does it support your objectives? Did you accomplish your objectives? Okay, based on your data. And then after you've based it on your data, okay, then what do other people say? Okay, how does your material compare to others? Okay, and so you can see that I have a lot of citations in my discussion on this topic because other people have written about it. Okay, and some of it supports my findings, and some of it does not support my findings. And my findings are differ from those of previous research. That's why you have a discussion. That's why you pull in your information, your statistics, your data, and then see what others say about that topic. Okay? Now, in some cases, it's not a perfect match. So maybe McCuncrick and Dixon, maybe they weren't talking about white suckers. Maybe they weren't talking about the species of fish that I was talking about. Maybe they're talking about trout, and they found something different than I did. Okay? Now you need to discuss that. Okay? Well, so even though my study might have found this, and their study found that, it was a different species of fish. Or, maybe you're looking at plants, and it was a different species of plant, or something along those lines. Okay? So that's the discussion. However long you need the discussion to discuss your hypotheses, the support for your hypotheses, and, the results of the information that's out there that either supports your hypothesis, so the literature out there that supports your hypothesis, or refutes your hypothesis. Either way, okay? Remember, it's okay to not have a supported hypothesis. It makes no difference if your hypothesis is deemed wrong, okay? In fact, majority of hypotheses are incorrect, okay? Once you collect the data, then you'll know if your hypothesis is correct or if it's refuted. Okay? So remember, your hypothesis is not proven. Your hypothesis is not disproven. Okay? It's either supported or refuted. Okay? So supported or not supported by the data that's collected. That's it. Okay? All right, and the last piece is the literature cited. Literature cited should be consistent. Okay? And I'll often have students ask, well, what kind of method? MLA, APA, Chicago, Well, actually, we use a method called CSE. That's what scientists use. But I'm not asking you to use it because it really makes no difference. If you know MLA, use MLA. If you know APA, use APA. If you know none of them, then look up CSE and use CSE. But be consistent. Okay? Alphabetize your literature cited or your reference section. It depends on what you want to call it. Okay? Alphabetize it. All of your cited resources should have authors. Think about it. If you're going to put a citation in that's supporting your hypothesis or refuting your hypothesis, but there's no author, you have no clue who wrote that information. I mean, that's like saying, I know I'm correct, but I don't know who told me I'm correct. Okay? I know this information, but I can't tell you where I got that information from. Well, no one's going to believe you. If you don't put a name to it, okay, then no one's going to believe you. So yes, you have to have an author. Yes, if you're using websites, you have to have an author. If they're not willing to put who wrote it down on the website, then you're probably correct in being skeptical on whether or not that information is true. Okay? So, if you're going to use websites, find the author, find the date it was published. Okay? Write down the title in which they published it under. Okay? And then if it's a website, of course, you can use the web address so I can find it. Okay? If it's a book, then I need, the title of the book, the editors of the book, etc. If it's a journal article, the title of the journal, the volume of the journal, and then the page numbers, etc. Okay? So, with all that being said, hopefully that clears up any confusion that you might have around writing a lab report. It's, it can be a daunting task, but you have a very long period of time to do it. Okay. It is due the last day of class. Okay. Which I will post a link the first week of May. That Wednesday is the last day of class. At least I believe it is. Okay. And I'll post that a link, and that's when it's due. And you just upload it to turn it in. Okay. So again, if you have any questions, let me know. But this is the formal lab report that you're going to do this semester. And you get to choose the lab that you're interested in, okay, and go forth and do good work. All right, till next time.